Hello and welcome to our second lesson on enzymes. I know in the last lesson we didn't really talk about why there was this lock and key picture at the start, but this is the lesson where we'll be looking at it. So after looking at what is a substrate and what are some characteristics of enzymes, we'll be looking at what we call the lock and key hypothesis of enzyme action. This really is the most important part of this lesson. So on the textbook, this is from page 77 to 79. So let's start off again with a simple reading task, just to get you guys familiar with your textbooks. Just have a read of textbook page 77 and answer the question, what are the characteristics of enzymes? Alright, so pause the video here, have a read and come back again. Okay, so the three characteristics of enzymes that you should have picked up from the textbook are: first, that enzymes speed up chemical reactions, they're required in minute amounts, and finally that they're specific in action. Alright, so let's go deeper into each one of them. Now I'm going to take the first two characteristics of enzymes together, that they speed up chemical reactions and are required in minute amounts. Now these two characteristics can be found or taken from the definition, which you should already be memorizing. So if you remember this definition, then you will remember these two characteristics, right? Because in the definition, it already says that enzymes speed up chemical reactions, right? So this is the same as the first characteristic. Secondly, in the definition, you remembered that enzymes remain chemically unchanged at the end of the reaction. Now to say this differently, you could say that enzymes can be reused, right? So they're used in one reaction, then they finish, and then they can be reused again. And that's why you don't really need a lot of enzyme. You just require a small amount of enzyme to act on a large amount of substance, for example, maltose, right? Because it can be reused again and again. So it's for the same reason that when you use something like biological washing powder, right, normally you just add a little, just 1 cup of washing powder into your big washing machine, right? So you don't really need a large amount of enzyme in order to wash the large load of clothes, because enzymes can be reused and they remain chemically unchanged at the end of the reaction. So I want us to spend a bit more time thinking about what it means for enzymes to be specific in action. And to start off, I want to think about an interesting question that is relevant for our lives, right? So sometimes you see some animals like cows or sheep, and they're just eating grass and they survive on that. They get energy from the grass that they eat. Now why can't we also get energy from grass like cows? Now here's a hint. It has something to do with the cell wall of the cells that make up the grass. Now if you're thinking about cellulose, then you're on the right track. Now as we learned in the last few chapters, plant cell walls are made out of cellulose. And the thing is, our digestive system has some enzymes, but the enzymes in our digestive system are not able to act on cellulose. They cannot break down cellulose. Now, on the other hand, cows in their stomachs have bacteria that produce an enzyme called cellulase, which can act on and break down cellulose. So that's why cows can get lots of energy just by eating the grass, but we can't do the same. Now the fact that the enzymes in our digestive system cannot act on cellulose or break it down is because enzymes are specific in action, or this thing called enzyme specificity. Now what that means is every enzyme can only act on a particular substrate. A substrate is the substance that an enzyme acts on. So for example, you have learned about amylase in Sec 2. Amylase is found in our saliva, it's an enzyme. Do you remember what is the substrate that it acts on? Right, it acts on starch, which can be found in foods like bread. It acts on starch and it breaks starch down into maltose, if you still remember. Now, the fact that amylase is specific in action means that it can only act on starch. It cannot act on other kinds of substances. So it cannot act on cellulose, for instance. And that's what it means by an enzyme is specific in action. One enzyme acts on one substrate and on no other. Now on a side note, the substrate, as I mentioned, is the substance that an enzyme acts on. So for the enzyme amylase, the substrate is starch, protease, substrate is protein, lipase, substrate is fat, and maltase, substrate is maltose. So just get comfortable with the use of the words enzyme and substrate. You'll see a lot more of it in this topic. Now coming back to enzymes being specific in action. Well, that's a lot like locks and keys. I don't know if you have this problem before, where you have a bunch of keys, right? And then you have one lock. And you're looking at that lock and you're wondering which key unlocks this lock. And you try every single key, but none of them works. And then you try again, and eventually you find the one key that works. You know, that frustration happens because locks and keys are very specific, right? One key unlocks one lock, and every lock is unlocked by a particular key. So it is here that we are going to talk about the all-important lock and key hypothesis. Now this lock and key hypothesis explains why enzymes are specific in action. And the lock and key hypothesis, well, it describes the action of enzymes on substrates as an interaction between a lock and a key, right? And that's why it's called a lock and key hypothesis. So the first major thing to note is that in the lock and key hypothesis, the enzyme is the one that acts like a lock, right? Over here, the enzyme is like a lock. Over here, I see the enzyme is the one with a kind of a depression inside. We'll talk about that in a while. And the substrate is the one that acts like a key. So this is the substrate, and this is the one that fits into the little depression or pocket on the enzyme. Okay, so just get that clear. People often mix it up. The enzyme is the lock, and the substrate acts like a key. So here's just the big picture of the lock and key hypothesis. Right, first the enzyme over here, right, it will bind to the substrate, which is over here, to form the enzyme substrate complex. Then the enzyme acts on the substrate, changing it into the product, and finally the enzyme releases the product. Okay, we're going to go into these stages into deeper detail. Okay, so just take note of how we describe the lock and key hypothesis. So when describing the lock and key hypothesis, the first thing you need to state is that the enzyme has a depression or a pocket, right, called an active site right here. This is the active site. It is the depression or the pocket, highlighted in red. So this active site has a specific three-dimensional structure, right? It's a very specific shape, as you can see. And secondly, you need to talk about the substrate, right? And the substrate, remember, acts like a key, right? And you need to say that the shape of the substrate is complementary to the shape of the active site of the enzyme, right? You need to know this word complementary. Okay, what it means is that it matches, right? So for example, look down here. Okay, you can see that the substrate has a shape, right, that is complementary or that matches the shape of the active site. So both are matching. And hence, the substrate is able to fit into and to bind to the active site. Okay, you need to know these words, fit into and binds to the active site. And as a result, we form a enzyme substrate complex. This is the enzyme substrate complex made out of the enzyme and the substrate bound or joined together. Now that the enzyme substrate complex is formed, the enzyme can act on the substrate and convert it to the product. So in this case, the enzyme is causing this substrate to break down into two other substances. And finally, the product leaves the active site, it is released, and note, the enzyme remains unchanged, right? And it can be reused. And that's why we need only a very small amount of enzyme to begin with. So a very common question that will be asked is, how does the lock and key hypothesis explain why enzymes are specific? Or the question might ask you, using the lock and key hypothesis, explain why enzymes are specific. Well, here's an easy way to answer such questions. So you can start off by saying that a particular enzyme has an active site that has a specific three-dimensional shape. Right, this three-dimensional shape is only complementary to the shape of, whatever substrate that is specific for it. And it cannot bind to any other substrate. Okay, so let me give you a simple example. For example, when talking about amylase and why amylase is specific, so let's say the question says, explain why amylase is specific. Then you can say that amylase is specific, right, because it has an active site with a specific three-dimensional shape. And that this active site has a shape that is only complementary to the shape of starch, right, which is what amylase acts on. So amylase cannot bind any other substance or substrate, right, like cellulose. So by saying this, you can use the lock and key hypothesis to explain why enzymes are specific. Now let me just end off by saying that the lock and key model has its limitations. In fact, there's another model of enzyme action called the induced fit model. Okay, it's totally not in your syllabus, but just something to be aware of. And the idea is that this active site is not entirely rigid. It's not entirely fixed in shape. And what people say is that when the substrate enters the active site actually changes its shape slightly to bind more tightly to the substrate. So a bit like this hand that bound, like grasp around the ball. Right, so that's the idea that some people have about enzymes, which is slightly different from the lock and key model. Alright, so just to get yourself familiar that not all models are perfect. Alright, so with that, end of this lesson, please get yourself familiar with the lock and key model before we go on to the next video. Great.